Well, we made it. You're still here. So we're going to go into, this was the last formal part. We still have a poster session afterwards where we will give out the awards to the winners of the Rimoine and Peltonen Awards. We still have a few surprises, but now we are going into the part where we're going to honor today's honoree, Lise Neufeld. So first of all, thank you very much for accepting to do this. It took a little bit of convincing. But once we announced it, as you can see, we got a tremendous response from people who really wanted to do this and wanted to be here today, including someone who traveled from St. Louis just to be here for you, and a lot of other things that you'll see along the way. I was hoping for this to be a bit of a roast, but giving Liz's personality, it sounds like it's going to be a hagiography the whole way through. But so please feel free to tell stories. To MC this part, Steve Suderbaum will be your person of choice.
Well, as Emmanuel said, I'm Steve Cederbaum, and I've been asked to be the moderator of this session in which we're recognizing Liz's great contributions to both science and to UCLA human genetics. I first met Liz when she arrived in 1984, and we have been friends and colleagues ever since, and we share our passion for opera, and we have the same series at the LA Opera and usually meet in the lobby afterward and give a very brief dissection of what went on. I'm going to give brief introductory remarks and then kind of moderate the session in which other people are going to say things in tribute and possibly roasting Liz. Some months ago, I finally got to read a book called The Eighth Day of Creation by Horace Freeland Judson, which, by the way, is, I think, the finest chronicle of the early days of the molecular revolution that's ever been written. And, of course, all of us are part of that revolution. Genomics and sequencing has played an enormous role in almost every discussion today, and this went on. But when I got to the part which is my own deepest interest, which is the regulation of gene expression, I was struck by several things. Mostly, it's the exquisite beauty, originality, and most of all, the simplicity of the studies. Their greatness lay in their conception and in their planning. The results were simple, they were straightforward, and they were often definitive. The cost of performing these studies was very low, and this really defined my notion of real science, where the design of the experiment rather than the gathering of data played a predominant role. In 1968, as I was beginning my fellowship in medical and human genetics, and yes, in those days, every medical geneticist was trained as a human geneticist, I encountered what I thought was just such a paper from the lab of someone of whom I had never heard. It was the first of 3 papers over 2 years that completely redefined our understanding of the operation of the lysosome and of the nature of lysosomal diseases. As each one came out, I was impelled to present that to the Journal Club at the University of Washington where I was, and we discussed their impact on our understanding of lysosomal storage diseases. You can imagine what a thrill it was for me to greet Liz and to meet her when she arrived at UCLA. If I had ever spoken with you before that, I'm really unaware of it. But you were, of course, a legend when you arrived. A brief biography. Liz was born in Paris to Russian immigrant parents. I should say that I derived this biography from something Liz herself wrote in the annual review of biochemistry in 2011, so I didn't have to go to Google or anything. I just had to ask Liz where to look to get a picture of her. And she came to the U.S. in 1940 by a then common circuitous route through Portugal. Any of you who have read World War II novels of intrigue know well about that pathway. After graduating from Queens College, it is fair to say, Liz, that you bounced around some in various science jobs, and during that time bounced into the arms of Ben Neufeld, with whom Liz has had 2 children. I don't know if that constitutes a roast, saying Liz bounced into the arms of Ben, but that may be as close as I'm going to get. She's going to have a chance to reply, so we'll find out. A not incidental aspect of this period was the fact that Liz faced overt discrimination at graduate school by decent and well-meaning people because she was a woman. This was a different era, folks, and I know the problems aren't all solved, but this was more extreme in those days. In any event, she entered a wonderful PhD program in biochemistry at Berkeley with some really legendary figures, but particularly in carbohydrate biology. And she describes this period in her short memoir as mixed and not exceptionally successful. In other words, Liz's great success did not begin on day one, and every struggling grad student should take heart and take note of this fact. But the common theme of Liz's scientific career was the study of the synthesis and the degradation of complex carbohydrates, and this work, of course, has continued to this very day. There were a number of significant characters in the carbohydrate field who crossed her path at this time: Vic Ginsburg, Rosalind and Stu Kornfeld. Probably most of the audience here doesn't know them. I did well, and they were just wonderful people. Liz became interested in Hurler disease in 1967. That astounded me. And what was most remarkable about this is that within 2 or 3 years, she made the findings and the discoveries that completely turned the field on its ear. She demonstrated first that these disorders were due to the accumulation of carbohydrate, not because they were oversynthesized, but rather because they weren't broken down. This may seem like an unremarkable finding today, but then we didn't have any idea what the biological basis for lysosomal storage disease were, and this was a significant finding and carried out by, again, these simple, straightforward, and definitive studies that I described. Following this simple model of defining experiments, Liz and her collaborator at the time, Joseph Frattantoni, had planned experiments in which cells of different types were to be mixed. However, the pivotal experiment in which Hurler and Hunter cells were mixed was the result of a laboratory accident. They were not supposed to be mixed, but in the true spirit of what is a brilliant science and genius is the fact that when you get something that hasn't worked properly, instead of just throwing it out, you look at it and say, what did this experiment show and what might I have learned from it? And what they learned from this mistake was that there was cross-correction of the cells. And this was quickly followed by demonstrating that the medium from these cells mediated, if you will, the correction, and eventually the purified enzymes mediated the correction. Ironically, this was the genesis of an entire billion-dollar industry. That simple experiment done in 1967 or 68 led to literally a billion-dollar industry, and Patty gave you a wonderful introduction to that, and you're going to hear more about that in a little bit. Needless to say, Liz went on to amplify these findings and to continue until this day to make a host of important findings in the biochemistry, physiology, pathophysiology, and therapeutic approaches to these disorders. And you'll hear more about that from Liz's colleagues. Before arriving at UCLA in 1984, sounds like George Orwell, and it was a kind of a revolution that was probably worthy of George Orwell. Liz sent advanced scouts, two talented individuals, one of whom is here today, Lenny Rome and Larry Shapiro. They foreshadowed what a Liz Neufeld recruitment might bring to UCLA in their demeanor, in their scientific brilliance. Nothing, however, prepared UCLA for the impact that Liz would have. She's a dynamo, a catalyst for change with no mutations, an innovative teacher with whom I had the pleasure to run a seminar, and of course a superior scholar and leader. Although she was the chair of biological chemistry, she is being honored today by human genetics. There is good reason for this. From day one, when Liz arrived, she teamed up with David Rome to develop this joint program, interinstitutional program for medical genetics, which has thrived so well until today. But more importantly, I started to describe Liz as the midwife for the birth of the human genetics department, but then that was really not enough. She was not only the midwife, she was like the mother and father and midwife. It's kind of, I liken that to something like Vishnu, the Hindu god with 4 arms, or a Greek mythological figure who was more anatomically and dynamically enabled than the rest of us. And she drove and willed this department into existence. I was amused when Wayne Grody introduced me as waving my arms around, and here independently I had written that allusion to Vishnu, although Vishnu usually is pictured with his arms not moving. Of course, during this journey, Liz picked up some minor accolades, like election to the membership of the National Academy of Sciences, receiving the Lasker Foundation Award, the Wolf Prize, and probably if I had worked at it, I could have come up with a dozen more honors. Liz teaches us humility with her own story and inspires us with her energy, insight, leadership, and above all, decency, all characteristics that she's modeled for us until this very day. So now we're going to begin the formal program with this introduction, and it's going to begin with Liz herself, who required the first word, and maybe she'll ask for the last word.
Thank you, Steve. So I thought I'd tell you a little bit about the early history of the training program in medical genetics and of the department, because I was quite involved in them, as Steve told you. So when I arrived in 1984, there had been a very good genetics program at Harvard, but it was scattering. Mike Kabak was going to San Diego, David Rome was going to Cedars, and the future of genetics was a little bit not known. And so I talked to David Rome, and I told him about the Inter-Institute Medical Genetics Training Program at the NIH, in which I had participated. Now, that's the program in which Ellen Zadransky had trained. And I asked, why couldn't one have an analogous intercampus program between Cedars and Harvard and Westwood? And Dave Rome liked that, and he started working on it right away. We then had to think about how to fund this program, where to get the fellowship money from. And David started working on applying to the NIH, and I went to Dean Sherm Melenkroff. Sherm Melenkroff had, in anticipation of my coming here, he had recruited me, and in anticipation of my coming, he had obtained some rather large grants from several foundations. And one of them, the Weingart Foundations, came in 5 installments of about $400,000 each, which at the time was an enormous amount of money. And discussing this with Chair Mellencroft, I was able to, well, we agreed that the inter-campus genetics program, genetics training program, would be a very fine recipient for one of the installments. And that allowed the program to develop until it became an NIH-funded program. And even after that, there was some money left over, and we could have extra fellowships and so on until the money ran out. So that was very nice and very simple. Getting a human genetics department was a little bit more complex. Now, there had been, for many, many years, a lot of people doing genetics on this campus, and specifically human genetics. But there was no umbrella organization for them to interact and perhaps have course together and have activities together. And because of many people asking for it, Dean Ken Shine put together a committee in 1989, and some of you had served in this committee and remember it. And the committee, which was called the Shapiro Committee, because Larry Shapiro was the chair, this committee deliberated long and interviewed a lot of people, had outside consultants, and came out with a report which recommended that there be a center with a small c, a genetic center first, and then after 3 years that might develop into a department. By now it was in early 1990s, and the state of California was in a bad way, and that meant the university was in a bad way. And so Dean Sidney Golub had to put a moratorium on this. And he was able to lift the moratorium and start garnering resources only in about 1993. Okay, so at that time, by that time, I think Larry Shapiro was leaving, and I took on the job of writing up a program for developing a department of human genetics. Many of you may remember this because I would bombard you with requests for letters that would, in which you would say that you were very enthusiastic with the idea of a human genetics department and that you would cooperate in creating it. By 1985, we had an application that had been approved by the departments and the department chairs and the faculty executive committee, of which Lenny Rome was then, I think, the head. And I forget the deans of the medical school and of the college. And we sent it off with great pride to the Council on Budget and Planning that had to approve it. And would you believe it, they didn't like it one bit. And I didn't keep the rejection letter, maybe it was too painful, but I found a letter in which I summarized the concerns of the committee. I summarized them for Dean Levy, who would eventually have to respond to it. So let me read you the concerns. Concerns number 1. This is a carte blanche for a new department without a clear sense of what the department would be like. Concern number 2, the goals of the department are not clear, and the method of ensuring that such goals would be implemented was not clear either. Concerns number 3 and 4, which are quite similar, it wasn't clear how the new faculty would interact, how the current faculty would interact with the new department. And it also wasn't clear whether they would be willing to teach in the new department. So what to do? So you have to do what you do when your grant application was rejected by a study section who obviously hadn't read it and certainly hadn't understood it. And you also have to provide new information. So I went with another round of letters that some of you may remember, which was that you would promise to teach, if you already were teaching a human genetics course, that you would continue teaching it. And if you were a chair, I don't see any chairs from that time here, but for the chairs and the heads of interdepartmental programs, that they would allow their faculty to teach for the human genetics department, and that they would give them credit for such teaching. So this went out together with some new information from Dean Levy, that there would be a new building, that it would soon be, the building would, construction would soon start, and human genetics would be given a big chunk of space in that building, the Gonda building, and that there would be some interim space in the Brain Research Institute. So this time, the application was acceptable to the Council on Budget and Planning. So there was another thing we had to do. We had to have a plan, an application for a graduate program. And new graduate programs have to be approved all the way up to the Office of the President. Okay? So writing up such an application wasn't very difficult. I used as template what biological chemistry was doing, and with appropriate changes. And we ended up, it seemed okay, but there was a problem. You couldn't have a graduate program without a department chair. And you couldn't recruit a department chair unless there was a graduate program in place, which was a bit of a catch-22. That was solved. And I don't know who proposed this, and it wasn't me, it was somebody who was administratively very clever, that the graduate program would be approved at that time, but it couldn't be activated. That is, no student could be recruited until there was in place a chair and two faculty members with a primary appointment that was tenured or tenure track. Okay, and with this, we had the program ready to go. Lena Peltonen was recruited, and as of July the 1st of 1998, there was a chair and there were two faculty members, and we recruited, the department recruited its first graduate student. And you know the rest of the story, which is a success story. So thank you for allowing me to tell you that.
Well, I think that the next speaker is going to be the person who was referred to twice, and that's Lenny Rome.
Thanks. Great. Good afternoon, everybody. So, you know, I was supposed to talk about Liz today, and after all the I've heard so far, that would actually be boring. So I'm going to instead change my talk, and I'm going to talk about my favorite subject instead, which is me. So what I'm going to present to you today is using the most convincing pseudoscientific evidence available that the Department of Human Genetics should in fact be honoring me today. So let's get going. So here I am in 1975. I was on my way to NIH, by the way, to find a postdoc, and I was visiting with my folks. See if you can figure out which one of these guys is my dad. And so I was stopping in Ohio on the way to NIH, and I had narrowed my search for a postdoc down to 3 individuals, Julius Axelrod, Roscoe Brady, and Elizabeth Neufeld. I hadn't met any of them. I had corresponded with them by mail. Remember, Al Gore had not invented the internet yet. And they were all willing to talk to me about a postdoc, so I threw caution to the wind, flew across the country, and met with them. Now, the first day of my visit, I met with Axelrod and Brady, and it was not a good day, okay? Julius told me that he had already filled up his lab, takes 1 postdoc a year, and said, you could wait a year and come work with me next year, but I was in a hurry. And then I met Roscoe Brady, and if any of you know Roscoe, he gives you a distinct impression. For me, it was an impression a cross between P.T. Barnum and Doc Emmett Brown from Back to the Future. So needless to say, I was really in trouble at that point, and I thought I was going to be leaving Washington without a postdoc lined up. But fortunately, my meeting with Liz went very well, and we agreed that I would start a postdoc later that year. So I looked around for a picture of Liz from around that time, and those of you who know Liz know that she does not like to get her picture taken very often, and so I had trouble, but I went on the internet and I Googled Elizabeth F. Neufeld, and I found this picture of her. You can see. This is perfect because I think this was the picture Liz used on her NIH ID badge, and you could tell why I was really anxious to get into the Neufeld lab. So I found another early picture of Liz from around, I think earlier than that time. This is her without her makeup on, so, but it's a real picture of Liz. And I want you to know that in contrast to that carefree, jocular extrovert that we all know her as, at that time, she was very serious. She was a very serious individual. She was working day and night, but occasionally she would take a break. And usually she would go on a hike on the weekend with her husband, Ben. And this is actually a slide from right about after I arrived at NIH, where Ben and Liz dragged me on one of these hikes. I want you to know that I plotted Liz's fame index against time. And I call this the EFNFA, Elizabeth F. Neufeld Fame Analysis. And because I wanted to show you that coincident with my arriving in the Neufeld lab in 1975, her career really took off. And this is really definitive evidence that I want to bring you. So I do want to show you a few photos of people that were in the lab at the time. I don't know if Liz remembers any of these pictures, but Aki Vastason, Gloria Sando, Inga Lebers, Jerry Garvin. The lab was so crowded, Jerry Garvin and I not only shared a lab bench, we actually had to share a lab desk at the same time. That's a true story. And then a lot of other people who I overlapped with in the Liz lab. By the way, down here, Paulette, Titus Dillon, Mike Zasloff, and Dan Camarini Otero. And in data that I really don't have time to present to you, but we can make it a supplement to the presentation, all of these folks, nearly all of them, became very successful scientists on their own right, and I do not think it's a coincidence that that overlaps with my being in the Neufeld laboratory. I'm just saying, okay? By the way, Liz never ceased to amaze me about her talents in the laboratory. As Steve said, we knew that she could speak French because she had been born in Paris. But one day, I was on the phone with a long-distance call, which at that time was difficult, with a Russian scientist who wanted to send some cells to Liz to analyze for one of the lysosomal storage disorders. And he was having trouble understanding how to mail the cells, how to fill the flasks. Liz was getting very impatient. She puts out her hand. She says, "Give me the phone." So she takes the phone and then goes into this fluent Russian, giving this guy a lecture on how he should know how to mail cells and do other things. And then the cat was out of the bag that she was multilingual. And I think the State Department heard about this because a short time later, this is the true story, they called up and they said there was some Russian scientists had come to the State Department with a diplomat, and they needed something to do with these scientists that evening, and could Liz entertain them? And she thought, well, that would be no problem. So she asked me to look in the newspaper to see what kind of entertainment was going on that day. And again, there was no internet, so I had to actually find a newspaper. And the only thing I could find that was playing, that was available, was a show of the Grand Ole Opry, okay? It's really true. And so Liz says, oh, that should be no problem. Get tickets to the Grand Ole Opry. And because it was downtown, she said, could you come with me? Can you come? I said, sure, I'll come. And so she hands me the keys to her Saab, and she says, you're driving. Now, anybody who knows about Liz's driving knows that this was a very good idea, okay? So we pick up these, 3 or 4 Russians, okay? And they were out of central casting. I swear, the head guy was Nikita Khrushchev with like indigestion. I mean, and we drive down there, and Liz is talking to them in Russian. They didn't speak a word of English, and they were answering in kind of grunts. Okay, they really weren't saying anything. And we got to the Grand Ole Opry, and we actually had like front row orchestra seats. That's how popular that show was that evening. And it was amazing because it was the worst show I had ever seen in my entire life. I'm serious. It was a combination of bluegrass and burlesque, and the whole time the Russians just sat there with their arms crossed and didn't say anything. And I'm thinking, this is an international incident. Liz is going to be thrown out of the NIH. The State Department's going to be embarrassed. It's terrible. I came in the next day. Liz is all smiles. Everything's fine. The Russian guy called her up, told her it's the greatest show he'd ever seen. How could she know that this was their favorite genre? It was absolutely amazing. Absolutely amazing. Okay, so a couple years after that, my girlfriend, Melanie, and I decided to get married, and we scheduled some wedding and a reception in some schlocky banquet hall in Silver Springs, Maryland, and we invited Liz. Now, up to that point, I had never seen Liz dressed up, okay? And I was so surprised when she arrived at the banquet hall that she must have been there before, because she came dressed to match the wallpaper of the hall. So here's yours truly, and Melanie, Liz, and Ben. Now I want to point out one more thing here. Okay, so this is 37 years ago. Okay, and Ben is already, his pants line is already creeping up. 37 years ago, and if we flash to today, you can see where Ben's waistline is, okay? And the reason I selected this picture is some of you did not get my driving joke earlier, okay? So that's the only reason why I picked this picture. Okay. So nearly 5 years after I joined Liz's lab, I decided to depart and take a job here at UCLA. In the Department of Biological Chemistry. And of course, I stayed in touch with Liz. And she visited UCLA twice during that time, once to give a lecture and once during the recruitment. And the first time Liz visited, Melanie and I had just bought a house in Studio City, and so we invited her to come and stay with us. I was a little nervous about that. I wanted to set a good impression for my postdoc advisor. And we had a little dog at that time named Stella. Actually, I have a picture of Stella here. And this is Stella and I sitting on the porch of our house. And Stella was kind of mischievous. She liked to get into laundry. And she would find socks and carry them around the house and play with them. And when Liz arrived and we moved her into the guest room, Stella became particularly annoying. And she came out from the guest room one day with one of Liz's socks in her mouth. Well, that evening we were getting ready for dinner. And I had cooked an elaborate spread to try to impress Liz. And just as we were getting ready to sit down for dinner, out from the bedroom area came Stella with a pair of white underwear in her mouth. And she walked right up to Liz, and Liz snatched the underwear out of her mouth and then panicked when she realized that it was a pair of my underwear. I'm not sure who was more embarrassed, Liz or me. So a couple of years later, we were in need of a new chair, and as both Larry Shapiro and I were faculty in the department, both of us had previously been postdocs with Liz. Here's Larry, by the way. We suggested to the chair of the search committee at the time, Irv Zabin, that they should contact Liz and see if she was interested. And Irv called up Liz, and she said, no, I'm sorry, I'm very happy here at NIH. I'm totally not interested. And but a few months later, I happened to be chatting with Liz on the phone, and she had mentioned that there had been a new crop of administrators at the NIH, and she wasn't very happy with them. And so, as serendipity would have it, that same day, I was walking down the hall and I bumped into Irv Zabin, and he said, oh, this chair search committee is stalled. I really feel terrible that Liz Neufeld wasn't interested because she would make a great chair. And I said, well, you know, Irv, you might want to try calling her again. And so he did, and she agreed to come and visit. That made me very encouraged because, as you know, Liz is a no-nonsense person, and she would not have come and looked at the job if she wasn't serious. And she came and a visit, spent a couple days meeting with faculty and chairs. And at the end of her visit, she met with Dean Sherm-Mellenkopf. And the way I understand it, it was a very brief meeting. Sherm said, and this is a quote that I remember, Elizabeth, what would it take to get you to come to UCLA? And Liz told him what she would need, what resources she would need, and Sherm stuck out his hand and said, done. And it was the shortest chair recruitment in the history of the School of Medicine. I was a bit surprised to find out later that Sherm didn't have all the resources that Liz required. Minutes after she had left his office, she called our acting chair, Don Glitz, on the phone and said, Don, how are you going to raise the necessary money to get Liz here? That's also a true story. So here's an early picture of Liz in the School of Medicine, also found on the internet. Better yet, here's the Neufeld lab in 1985. Again, in keeping with my credit taking for this affair, Liz became chair of our department, was chair for 20 years, and all of the faculty on this slide were recruited under her tenure as chair. So again, I take credit for the success of the department. And as you already heard from Liz, that when she was forming the Department of Human Genetics, I was part of the faculty executive committee, and then later when the department was approved, I was in the dean's office. So I think it naturally follows that the Human Genetics Department should be honoring me for my role in the creation of this terrific department. So I'll leave with this really nice slide that we have of Liz. I'd have to label it, though, with a tribute to Lenny Rome's most distinguished postdoc advisor. So thank you all.
Well, that was an unexpected pleasure, and but I think he completely screws up the time schedule, but that was so wonderful it doesn't matter. I guess I'll go with the order that I have on my paper. The next person whom I'd like to call on is Catherine Ponder, who came all the way from St. Louis to be with us today and to celebrate. Catherine?
Well, that's kind of a tough act to follow. Now, if the truth be known, I'm from St. Louis, home of the St. Louis Cardinals, note the red. And when I heard about this, there was some thought, well, maybe they'll be playing Game 5 of the series, which would have been last night. So that was, and perhaps an impetus to come. But really, the true reason that I came was to give tribute to Liz, who I think is one of the greatest scientists. Now, I met Liz in the early 1980s, and actually I was a medical student at Washington University in St. Louis, home of the St. Louis Cardinals. And her son was a classmate of mine, Alice, and she came to visit and gave a seminar. And that was sort of the last I saw of her. And then after I graduated, I turned to gene therapy and I worked on hemophilia. And then for complicated reasons, I became interested in gene therapy for mucopolysaccharidosis. And at that point, started working on MPS 7, which is Sly syndrome. By the way, Bill sends his regards. And then ultimately worked on MPS 1, which is Liz's disease. I don't care what anybody, this Hurler guy, I think it should be Liz's disease. And so this led me to review her previous literature, and it's just an amazing story. She identified the etiology of Hurler syndrome, contributed to understanding how lysosomal enzymes traffic to the lysosome, and then developed a therapy for, or at least contributed to developing a therapy. And that's just an absolutely amazing story. And I think there are two traits that I think Liz has. One is that she's curious. I mean, how many of us, if you had mixed up these two cell lines and they looked normal, how many of us would have pursued that? And I think that's what characterizes a great scientist, is saying, wow, boy, isn't that interesting? What could be the cause of that? And second is her tenacity and attention to detail. So I called her up. I was working on gene therapy, and we started on MPS 1 in the early 2000s, and we wanted to purify that. We were working in dogs. We had the dog gene, and we wanted to purify the dog enzyme to use for looking for antibodies, and we couldn't get activity. And I called up Liz, and we had this 5-minute conversation, and she said, oh, well, you got to grow the cells in low pH, which makes a lot of sense, and lo and behold, that was correct. But this is somebody that knew the details of something she was involved in 20, maybe 30 years ago. Now Liz has 2 children, which she raised with her husband Ben. Sophie, her daughter, loves horses and is involved in teaching dressage. And Ellis, my classmate, got his MD PhD from St. Louis, from Washington University in St. Louis, home of the St. Louis Cardinals, and is now on the faculty at Harvard University in pediatric hematology. And I asked Ellis for comments on his mother. And actually, I do recall from when I met Liz in the early 1980s that Ellis described a really big Saturday night for Liz. And that big Saturday night was to get to drink a glass of wine while she reviewed and usually rejected JBC papers, as opposed to during the week, she just would review JBC papers without the glass of wine. So Ellis wrote this, and I've shortened it a little bit because of that, that loudmouth, on and on guy that preceded me. I did shorten it a little bit, but it reads from Ellis. Hi, Mom. Sorry I can't be there in person to celebrate your career. Of course, Liz never told her son that she was going to get an award. He might have tried to get here, but getting here from Boston is not trivial. Let me recount how great it is to have grown up with a distinguished biochemist and geneticist to chat with at dinner every night. We talked about all kinds of science over dinner, and I took that for granted. We talked over dinner about, let me get my glasses on, about non-random exon activation and the lion hypothesis, which got us into a discussion of calico male cats and XXY genomes. It got a little arcane. It drove my sister Sophie, the one who likes horses, crazy. As you know, she's not a science type. She used to moan, no, not again, and Dad didn't moan about it, or not much that I can recall, but it was probably tiresome. During college, I spent summers working at the NIH with Ira Paston, and one summer I worked on a mutant fibroblast line that couldn't glycosylate proteins correctly. In particular, it didn't have the N-acetylglucosamine, and my job was to prove that it was missing glucosamine phosphate acetyltransferase, the enzyme that makes the sugar that you yourself, Liz, studied 10 years before. So each evening over dinner, I would tell you the day's lab progress, and you would make suggestions about why the assay wasn't working, and I'd go out and try them the next day. And in the end, I got my first paper published, and it crossed my mind, which I later confirmed, that what was going on here was that Ira Paston was getting your expertise for my $6 an hour salary, but that turned out to be a good deal for everybody. And then he goes on, I skipped a little bit of stuff, but since Dr. Sadransky is the keynote speaker at the genetics retreat, he wanted to add a recollection about a Gaucher disease angle. And in February of 1983, there was an episode of the new hit TV show St. Elsewhere, and it took on a lysosomal storage disease. The young intern on the show was confronted with an elderly Jewish gentleman with nosebleeds. After hitting the textbook, she rightly reasoned that he could have Gaucher disease, and the drama turned on whether it was worth the cost of the expensive blood test to find this zebra disease. The older, experienced physician counseled not to do it, but of course the intern was proven right. As a med student in St. Louis at the time, I thought this was very cool. But when we spoke shortly thereafter on the phone, you were quite irritated that they had mispronounced the disease as Goocher, like the college in New York. Anyway, so Ellis writes, congratulations on the occasion of this celebration in your honor, and thanks for teaching me so much. And then finally, I'm actually part of the Division of Hematology in Washington University, and Stuart Kornfeld says hello, and Larry Shapiro says hello, and Scott Hickman says hello. And I would like to say that when Rosalind Kornfeld, Stuart's wife, died, we received donations and started a lectureship for distinguished women in science. And Liz Neufeld was our first speaker, and really is a reflection of the truly great women in science, women scientists. So thank you for letting me speak.
After all of this, I want to go off non-script. Ben, where are you? Do you want equal time?
Oh, sure.
I have to say that this is going longer than we anticipated, and is absolutely much more fun. I can't imagine even wine being this much fun.
I'm sorry I have no handouts. I was not prepared to speak until about 40 seconds ago. I don't even have a sign-up sheet where you can sign up and get the remarks later on. Liz's interest in the mucopoly, well, let me go back a way. When we were married, we were living in Maryland, and then we both decided to go to graduate school in Berkeley. And after a few years, Liz decided to go to NIH, and after a few more years, Liz decided to come here. So we have been yo-yoing back and forth across the country. So I know absolutely nothing about what happened in laboratories anywhere at either end of the country. I can tell you this. She came home from work one day, and after dinner she said something like, I read a very interesting paper today. The guy did, she mentioned the journal, I don't remember what it was, probably JBC. But she said, this guy did very good work. The only thing is that his conclusions are not supported by his data. Now wait, how can this happen? Well, I don't know, but she explained to me this controversy, I guess you'd call it, about what the mucopolysaccharidoses really were. And she said, this guy took one position, and the other position is this. I'd like to work on that. I said, well, go talk to your boss, who was DeWitt Stetton at the old Arthritis Institute. She came back a few days later and said, I did talk to him, and he thought this would be a good subject to work on. He said, wind up what you're doing, so that somebody else someday could pick it up and go to work on that. And so she did. And I didn't follow this. She came home one day, I don't know, years later, months later, and she said, Clara came in to my office today, and she had 2 of these bottles in which they'd been growing cells. They were using these up, and what they did when they used them up was pour all of the samples from one individual back into the same bottle. They had all grown identically, of course. And gradually they worked on what they had. But Clara came in and said, I don't know how I did this, and I'm quoting Liz or paraphrasing Liz. She said, both of these are labeled, they're from different patients and they're from different diseases. Should I throw them out? And Liz said, no, leave it alone, grow it, and let's see what happens. Okay. So few weeks later, she came in and said, something very strange. These 2 diseases cured each other. And I suppose if you had poured any 2 other diseases' cells together, this wouldn't have happened. She went on from there, came back one day and said, we had been working on a therapy. We have made a molecule of it. She came back later and said, we had this dog we tried it on, nearly killed the dog. Back to the drawing boards. The next thing I know, she invited me to come down with her to Harbor one evening, where they had bottles and bottles of a therapeutic, I guess you'd call it concoction, and they kept producing this stuff, and eventually it became of something. And this is very nice for me, because I get to go to all these parties where they celebrate advances in the industry. And here I am at another one today. Thank you all.
Well, we've been hearing from people all of these interlocking relationships that have gone on. It's something that's worth thinking about for the younger people here. But what was unsaid is we all knew one another and intersected along the way in ways that one couldn't imagine happening today, but what happened back in those days. Lenny pointed this out. And so the next person is going to fit into this theme. Ann Garber Ramoyne is here and wanted to say some words. And of course, you've heard about David and meeting with Liz and developing this program. And Ann, do you want to come up and say a few words? Ann and Annie Ramoyne is here as well, fresh back from Africa.
Well, this is really a privilege to be here with all of you, and especially to honor Liz Neufeld. My former career was as a genetic counselor, and I can remember way back then learning about biochemical genetics and being so impressed by this brilliant woman, and I was so happy that she was a woman and sort of stood out from all of the other investigators that I read about. And lo and behold, I ended up marrying David Ramoyne and then had many opportunities socially to be around Liz, who I was still so in awe of and got really shy around because I just thought she was so brilliant. But then as time went on, I really, through David, got to understand a different side of her as a collaborator, as a partner, as somebody who was willing to work outside of her immediate circle to build something bigger, to build something larger. And he always thought of you like that, Liz. And I really want you to know that he looked up to you as a partner and somebody who he could depend on for the greater good of medical genetics. And thank you so much for playing that role, and congratulations today.
Well, there are some other people who wanted to speak as well, and we're going to see a special treat at the end, so nobody leave. Now, I know that Audrey Fowler wanted to say a word or two. Audrey, and she'll introduce herself and her role in the life of biological chemistry.
A long life. So I want to thank Liz for coming to UCLA in 1984. She's always been a big help to me, and of course I was here long before she was here. And you saw the picture of Herb Zabin. Oh, sorry. You saw the picture of Herb Zabin up there, and he actually brought me back to UCLA, and we sequenced a long protein called beta-galactosidase. It was the longest sequence of a protein done chemically. It was 1,023 residues. And when we finished in 1977, what to do? So actually, the molecular biology invited me to set up a protein sequencing facility, which I did. And when Liz came in 1984, she said, I want you to move back to biological chemistry. And so she was really very supportive of the sequencing facility and me, and I thank her for that.
The next person is a reprise of this morning's session, but Patty Dixon wants to say some more words. Patty?
Thanks, Steve. So we've heard about how much Liz means to UCLA and means to the intercampus program, but I want to express how much Liz means to the lysosomal storage disease community. I also want to mention that when I was a first-year medical student, I learned about Liz. And I remember specifically the room I was in, everything about learning about Liz's discovery, and never thought that I would be here today. And so for the trainees here, you really have chosen a field where you can walk among giants. And what I've learned about walking among giants is that they learn as much from you as you learn from them. So I have collected a few notes from some of the people in the lysosomal field for you, Liz. First, David Begley from King's College London. Elizabeth is a seminal leader in the treatment of LSDs, and her demonstration of cross-correction opened the doors to enzyme replacement therapy and also the understanding of the mechanisms involved. This insight has been the torch that most of us have followed in later years. Her work has led to immense improvements in the treatment and quality of life for many patients with LSDs. I have met her on a number of occasions, and she is inspiring to speak with and still a great contributor to further progress and to the generation of new ideas and projects. Without her, many of us would not be in the position we are today. Treatment of the brain is now a great focus, and her leadership and foresight is without doubt in the field and is greatly appreciated. Her tribute is immensely deserved. It is for me a personal honor to know and have met her, and she is a giant in the field. From Haiyan Fu, Nationwide Children's Hospital. Dr. Neufeld is certainly one of the greatest as a scientist and as a person. Here's what I would like to say, hope it is not too Chinese. Thank you for giving me the support, help, and encouragement to stay focused on what we have to do to benefit patients and advance the science. You are and always will be my inspiration and role model as a scientist and as a person. Cheers. From Andrea Balabio, Telethon Institute of Genetics and Medicine. With her exciting discoveries, Dr. Neufeld profoundly influenced my choice to work on lysosomal biology and on lysosomal storage diseases. Since I was a student, I have learned so much by reading Dr. Neufeld's papers that I consider myself one of her trainees, even if I have never worked with her directly. From Alessandro D'Azzo, St. Jude Children's Research Hospital. I do not think it is necessary to mention all of Liz's scientific achievements and prizes, how much she has contributed to the LSD field and to the development of therapies for these diseases. I rather say a few words about my personal experience with Liz and what it meant for my scientific upbringing. I always considered Liz my mentor. The years I spent in her lab were among the most enjoyable, constructive, and serene of my scientific career. Liz has had a great impact on the way I think and approach science. Not only has she taught me how to design and perform experiments, but also, and most importantly, how to interpret and scrutinize the results and put them in the correct context. Her genuine interest in her work, broad and eclectic knowledge, and work ethic have been difficult to emulate. Although as a young postdoc, I was at times a bit intimidated by her, I learned how to read her expressions. I knew when her thoughts were no longer with me and she lost interest in my story and was already thinking of something else. It was always a challenge and a learning experience discussing work with her. Getting older, I often think back in a nostalgic way about those times in her lab in the decrepit Building 10 at the NIH, where she used to pin the best-looking audio radiograms on the wall of her office. Often late at night after a day of work, I remember sitting, waiting for the elevator to go home and just feel happy. Thank you, Liz. You will always occupy a special place in my heart, and of course, congratulations, yours, Sandy. From Professor Michael Beck at the University of Mainz. Dear Liz, congratulations. You have indeed deserved this award. I remember very well when I first met you the first time at the International MPS Symposium. I have learned a lot from you, and at the several conferences I meet you, I am always impressed by your cordiality, brilliant presentations, and discussions. From Barb Whittahays, Executive Director of the National MPS Society. Dr. Neufeld's contributions to the field of MPS resulted in treatments for these devastating diseases and the promise of hope for patients and their families. Her work in the field continues to reveal promising findings, which hopefully will lead to further treatments, especially for patients with neurological components of the disease. On behalf of the patients with MPS diseases and their family, the National MPS Society congratulates and honors Dr. Neufeld. And lastly, from Mark Dant, Volunteer Executive Director of the Ryan Foundation. In September of 1991, our family's future was forever altered when our 3.5-year-old son Ryan was diagnosed with MPS I. The news of his diagnosis and certain early death brought about many changes in our lives. The greatest of these changes was the pain of losing the gift of tomorrow. That day also brought us the news that there was little science being conducted anywhere in the world that might one day benefit our little boy. After crying for nearly a year, we began searching city libraries for anything published regarding MPS. It was then that I first found the name Elizabeth Neufeld because it seemed the majority of what was known about the biology of MPS was authored by Dr. Neufeld. 2 years later, I was honored to meet this great scientist. That conversation turned out to be the turning point in Ryan's future because she explained that the science behind a treatment for MPS I was ready to be developed and that she knew a young scientist who believed he could do it, a former student of hers, Dr. Emil Kakas. What I would later find out is that it was actually Dr. Neufeld's science from many years before that Dr. Kakas would be bringing to the world as a treatment for MPS I. Dr. Neufeld's life work has been dedicated to the purity of great science and what remarkable changes can occur because of it. Her dedication and legacy will forever be witnessed literally around the world in the smiling faces of those who are now living their lives with the promise of tomorrow because of her life's work. Those like our son Ryan, who once again dreams of what might be tomorrow. Thank you, Dr. Neufeld, for giving us the will to hope and the gift of our tomorrows.
I think Harvey Hirschman would like to say a word.
I actually hadn't planned to say anything. I just came to the festivities and to honor Liz. But it took me back and things because I tell people I've been at UCLA since right after the earth cooled. And I was here when Lenny was recruited, when Larry Shapiro was recruited, and when Liz was recruited. And based on today's performance, 2 out of 3 isn't bad. I actually had the pleasure of roasting Lenny at a party for him. I think it was his 70th birthday or 75th. I can't remember. But what I did want to tell you was that it was a different time when Liz was recruited, and I want to tell you something that I'm nostalgic for, and which I hope you'll find amusing. But I remember when we went through the process of recruiting Liz. I was on the faculty. Faculty's supposed to vote about things like this. And Liz and Sherm agreed on the fact that the offer would be made. And right then and there, they'd agreed on the fact that, yes, she was going to take the job. So Irv came to our next faculty meeting very chagrined and saying, well, Sherm offered the job to Liz, and Liz accepted, so now you have to vote on it. And I'm pleased to tell you that the vote was unanimous. And that wasn't because Irv was that intimidating. It was that Liz was that intimidating. That was, it was a pleasure to have Liz as a chairman. Liz is one of the straightest shooters, one of the fairest people, one of the most committed people. Actually, I actually want to ask you a question. I've been told a story that when, among the things that you told Sherm you wanted were many things, but you didn't ask for what normally happens, which is to get a chair stipend. The story I've been told is that you didn't get a chair stipend, and one of the reasons was that you just didn't want to be beholden to anybody about anything in order to make the best department you could with no conflicting interests. Whether it's a true story or not, whether it's apocryphal or not, I think it really reflects Liz's character and persona. She was always extremely straightforward. I did learn how to play her, though. I have to tell you that.
Now you tell it.
Oh, now I can tell her. At the time, I certainly couldn't tell her. Liz often is skeptical on first approach. I see Lenny nodding his head. Okay, and a lot of times she'll say, I don't think so, when you approach her at first. And so I learned to go to Liz when I had an idea about something and say, look, I want to propose something. I want you to think about it a little bit, and then I want to come back tomorrow and talk to you. And by then she'd had time to digest things, think it through, and inevitably, I would say that Liz, I mean, sometimes she would agree and sometimes she wouldn't, but when she wouldn't, she always had the right argument, and she was always on the right side. And she always presented things logically and forcefully, and not emotionally, but clearly and scientifically. It was always a pleasure to have her as my chair, and I think I can speak as one of the people who's been around the institution now, longer than most people, looking around the room, and it was a delight for 20 years to have Liz as chair. Thank you.
Well, now we're going to open the floor to anyone, unscripted or not, who wants to say a word. Just come up here if you like. We have no takers, so we have one final event that Emmanuel is going to put on, or someone's going to put on.
We first have closing remarks by our vice chair, Stan Nelson. No, you talk first while I install that thing.
Very good. Well, I'm just going to add my comments and my thanks and my admiration for you, Liz. That's some part of my closing comments. So before my role as dry vice chair of Department of Human Genetics, but you know, in 1993 when I came to UCLA, I was a bit of a fish out of water, working in genomics, genomic technology, having a footprint in the clinic, and very much wanting a basic science laboratory. And I'm very much indebted to Liz, creating a home where I was very welcome and a basic science department where I was welcome and allowed to flourish over the course of a few years. Although I was a little bit chagrined that as soon as the department occurred, I think Liz was very happy to get rid of me and push me off to human genetics. But thank you very much, Liz. Really much appreciate your support. So in closing comments, we're going to get a movie here after I'm done speaking, so don't leave. I just wanted to say thank you all for coming. This has been a great showing. I think it's worked out very well that the Medical Genetics Training Program, the Intercampus Medical Genetics Program, including Harbor, Cedars, and UCLA, along with the Department of Human Genetics, have fused together to make this retreat work, and it works very well. I think Liz's work has just been amazing to see. It's a spectacular career to see over the course of, 30 to 35 year, 40 year time frame of moving from simple observations, the experiment that you performed, that you followed up on, leading to actual meaningful therapeutics in kids with rare diseases. There's thousands of different rare diseases and hundreds of therapies. There's a massive amount of work that we have to do to solve some of these really catastrophic illnesses that occur primarily in children. And I think what you've done has laid a foundation for that. I think the fact that there's a multi-billion-dollar industry around this is really spectacularly related to the enzyme replacement therapies, and that creates an economic framework by which these therapies can actually move forward. So thank you very much from a number of perspectives. I also think that bringing the two programs together does a very nice job of sort of bringing the issues of what happens in the clinic back to basic science questions and what happens in our basic science laboratories to impact the clinic. And I think those things are fusing very nicely here. So I'd just like to congratulate all for their great presentations. And I think you're going to have to push this.
Liz, sorry I could not be there for your tribute because of work taking me abroad. I knew I'd have to say something about the mentor, scientist, and friend that led me to the biotech business that makes me so busy that I have to go abroad so often that I have to miss your tribute, despite the scheduling gymnastics the organizers went through. So for this tribute on video, I thought about talking fondly about the deepest, darkest secrets that make the Liz we know tick. But then I realized I don't know any. Fact is, Liz is a pure scientist, unassuming, unpretentious, and focused on getting the truth about how things work and why. When she started at the NIH in the 60s, she read and read, looking for the question that would occupy her career, and the MPS diseases were just there, akin to the carbohydrate chemistry she knew, but also the cause of a disfiguring and disabling disease of children when their genetics were just unlucky. And from the mindset of finding the truth, an accident led her to discovery the existence of a corrective factor. It was a bizarre stroke of luck that the cure was found before the disease was understood. And the decades after were spent figuring out why. A long parade of fellows came through the lab, of which I was at the end, the last runner in a multi-decade scientific relay race. And getting there to treat the first patient was the first opportunity to change the medical future of many MPS patients.
If the treatment is successful, I think it will very much improve the quality of life for the patients. Today is another milestone in what has been a long climb. I don't think we're at the end of the road yet, but I think this is a very, very important milestone. I think this is one that I've been waiting for not quite 30 years, but I would say from that first experiment in 1969 where we could cure our cells in culture, that's 28 years. But then, of course, in medical research, that's not a very long time.
When I first met Liz to discuss working with her as a fellow, I had the pie-in-the-sky, unrealistic dreams of most postdocs, thinking we can figure out something like gene therapy and fix all these kids and go on and construct this great scientific career. But at our first meeting, the direction changed, and she said to me, "Emil, there are a lot of people working on gene therapy. I would rather just like to see us try to do the enzyme therapy since we've never had the chance to do that before." And frankly, I was a bit dumbstruck because enzyme therapy was really about making enzyme, which seemed like, an industrial project and hardly the place for a real scientist to waste his time. So I put on my poker face and told her, "Liz, fine, I'll do that for a couple years and then I'll get back to gene therapy." And let's see, well, it's been about 23 years since then, but I'm getting back to it eventually. So I started my research with Liz in the poor tropical outpost known as Harbor UCLA in Torrance. And there I struggled to make an enzyme that really did not want to be made. I found some old equipment and painted a lab and started making some enzyme. Now, one of my fondest memories of that old time working in the lab is that the cell line got made and the culture started producing enzyme. I'd come into the lab and tell Liz how much enzyme I'd made, and I could thrill her with that. I first told her the cell line was making 5 micrograms per ml, and she'd marvel that it took them 5 years to purify 5 micrograms. Then later I'd tell her that 1 plate of cells was making enough enzyme that she would originally purify from a 55-gallon drum of urine. That 1 plate had that much hyaluronidase content. And then another time I'd tell her I'd made 10 milligrams of hyaluronidase, and she was always amazed. And with the system shown here, I was making around 100 milligrams or so, and now enough to treat dogs with MPS I. And she was thrilled, as always. I made her a special vial that said Neufeld on it, thinking ahead for what was coming. But Liz worried and felt that no one would care about this rare disease and all these rare patients there wouldn't be a chance, and we might not ever get it developed. We went around together and we talked to people and companies, and frankly, no one cared at all, so it seemed like we were sunk. But fortunately, a family foundation working to save their child found Liz and soon was funding the work. Once we had seen Ryan Dance shown here with one of his famous pitcher stars from the Rangers, we both knew we had to do something for him. And they started raising money for us. And this is a picture of us with the first check from the Ryan Foundation. Now, we struggled with the support of the foundation, and eventually that special day came, partly with the support of a company. But that day was particularly special because they had the opportunity to give something back to Liz, and that is starting the first infusion that would be the first correction experiment in patients with MPS.
Ready to go? Good. Well, we're gathered for a very important occasion, a long time in coming, and I wanted everybody to be here because it really is 30 years in coming, and it's a special day for a lot of us. I think this journey began 30 years ago with Dr. Neufeld's lab with Dr. Frazzantoni, and then they, in an inadvertent mistake, mixed up some cells, and in that small mistake discovered how they might correct this disease using normal enzyme to get taken up by cells that don't have that enzyme. And took Dr. Neufeld and many fellows numerous years to go through the enzymology, the biology, cell biology of this disease, cloning the enzyme 1991, and finally producing enough recombinant enzyme to be able to administer it today to Justin. First dose.
Okay.
There it goes. It's going. That's it. Great. All right.
That moment was captured in this photo is really one of the most amazing moments right after the first infusion. And at that time, I gave a tribute to the work Liz had done to get us there.
It starts with a patient 30 years ago and takes this long path through cell biology, molecular genetics, and then back again to the patient. It really is an amazing story of scientific discovery and persistence.
If the treatment is successful, I think it will very much improve the quality of life for the patients.
I think what these 10 kids and families did is so inspiring and amazing and courageous, because they were really the first to step up to the plate and make things possible nobody ever thought would be possible for a future.
Emil gave us a different journey. We look back and we see the smiling faces of those 9 families and that picture of Emil in the director's chair. I would bet there isn't a day that's gone by since that picture was taken that I haven't thought of that. Such little kids and Greg. Heroes, all of them. 11 people in that picture, every one of them, heroes.
Now, the greatest gift I received from Liz was a chance to sit here with all my MPS I kids, young and old, to celebrate the completion of the 6-month study on the Queen Mary, and to realize that I didn't really want to be a basic scientist, but someone that made therapies for rare diseases. Because once you press that button and start infusion, you're forever addicted to that experience, and you want to do it over and over again. And since that time, the progress has been amazing. Ryan has done well with more than 16 years of treatment and now going to college. More than 1,000 MPS patients have received the enzyme, and the cell line made in that bungalow with the CNA from Liz's lab is producing that enzyme for the world. And from the success of that development have been treatments for Meritola Me, then Hunter, then Morchio, and finally now I'm working on MPS 7 with the team at Ultragenyx for SLY syndrome. Now, how likely was SLY syndrome ever to be treated? Extraordinarily rare disease, but my team and I have had the chance to take that enzyme therapy to that rare disease too. And I had the great thrill of being there again for the first infusion of MPS 7, SLY disease, with Dr. SLY.
Takis and I had a rehearsal for this about a dozen years ago. We proved that we could do it. Together we got funding from the government to develop the enzyme, but there weren't any commercial takers at that time. His company had too many other things on their plate. But with his new company, with its mission to treat diseases that no one else will treat, this really, and he always had kind of an emotional attachment to that disease, and we had a great friendship. So it really led to this day. I've been blessed in so many ways with a great career and a great family and a great wife, and now this is really a culmination of something I never really thought would happen because we tried so many years and couldn't get it to go.
Special day for Matthew, but really for all of us. We've been working so many years to get this opportunity today to treat Matthew. Dr. SLY is with us today. 40 years ago, they published a paper figuring out what was going on in patients like Matthew. 20 years ago, they figured out that maybe you could treat this. And I only got involved in the last few years and with the company to actually finish the job and take that science and make a medicine. And after a couple years now of work, we're now ready to give Matthew the first dose ever of enzyme replacement therapy for MPS 7. You want to say a couple words about this moment for you? You've been working on this for so long.
Yeah, it's really a thrill. I had almost given up hope that this would ever happen because it really took a special combination. I'm so grateful to Ultragenyx, grateful to Jeff Grubb, who was with me so many years and then joined Emil and that team to help make this possible. And it's really a fulfillment of a life's dream. I'm very happy to be here.
In honor of your work in this, start the first infusion. Where's the button?
Here we are.
It's going. All right. Congratulations. It's going.
So Dr. SLY and so many others have had a chance to see their work transformed into therapies for kids. And for me, the opportunity to get 3 products approved for rare diseases, to have developed several more of these products, and now to have founded a company dedicated to the same cause for ultra-rare diseases, with nearly 100 staff on 5 different rare diseases. It's a thrill I could not have imagined, Liz. Liz, I just have to thank you for directing me that way to the boring enzyme production project that no one cared about, which allowed me to see how much treating patients for the first time can make a career, which now everyone and their local venture capitalist seems to care about. I tried hard to think of the best possible tribute for you and wondered, what could I do that would demonstrate our commitment to the cause of transforming science into medicine and treating kids with rare diseases? How could I also give you that special thrill again, as I had in those early days as progress was made, and show you how much enzyme I was making? And I realized perhaps that there was something I could do that would demonstrate my commitment to your principles of great science and provide just one more opportunity to thrill you here in this tribute. One more opportunity to show you how much enzyme we can make. That's right, I have here 5,000 milligrams of iduronidase, or almost 2,000 vials worth $2 million. And I'm going to take the iced water iduronidase challenge. Right here for you, Liz. All right, Chantaria, let it rip. Liz, congratulations. It's tasty.